Okay, and the last talk for this session is by Jian from Harvard, who will tell us about decoding and translating the languages of biology. Okay,
thank you. So biology is multimodal. It's a symphony of different kind of modalities across, modalities, scales, and different languages. But for decades, we've been studying these modalities in isolation. There's a lot of advancements here, especially in this campus, that people develop a lot of cool microscopy tools. And then there's also, outside of this campus, there's also a lot of efforts trying to characterize the cell phenotypes through single-cell genomics. And actually, cells were discovered through microscopy. But there's always different kinds of, pros and cons with the different kind of data modalities. But the question that we wanted to ask is, how do we actually combine the best part of these two different measurements and then build a multimodal view so that maybe we can have a better understanding of the cell physiology across scale and across modalities on a larger scale and more efficiently? So how do we do this? So the solution seems simple. Let's just combine them together. But in reality, it's actually very, very difficult because one of the problems is for single-cell sequencing technologies, the information you can get is extremely high-dimensional, right? So you have 20,000 genes, let alone all these combinations, and there's chromatin, regulatory elements that's even way much larger than you can measure, than the gene expression. On the other side, if we're talking about the pixel size per measurement of the microscopy, usually it's very low. But if you combine all these different kind of pixel informations, you can get actually high-dimensional measurements. But on the other side, that there's also microscopy technologies that can give you high-dimensional measurement at the per pixel level. So that's why we're very excited about this direction, that maybe we should just start with the microscopy or imaging modalities that can generate one of the highest dimension that you can measure from a biological samples. So that's why we developed this method called Raman to RNA as a beginning of this journey. That this is in collaboration with Aviv and Peterson and Tommaso. We developed this method called Raman to RNA. The simple idea is that if you take a picture using Raman microscopy, can we generate single-cell RNA-seq data from Raman images? The advantage of doing Raman is because it belongs to a new, I would not say new, but it's a new category for biology that is chemical imaging. Because this chemical imaging modalities can give you a chemical activities at a high dimension in non-destructive and neighbor-free manners. So which means that at the per pixel level, you have up to 1,000 features, 1,000 channels that gives you the information of lipid, protein, nucleic acid, or different metabolite. So it's a composite measurement of the bioreaction inside of a cell. So combined with this, you can do, we already showed that in the paper that you can predict single-cell RNA-seq, but there's one caveat. Raman is one of the chemical imaging, but it's very, very, very slow. So which means that if you think about the scale that we're trying to do to map the human physiology, that is just not feasible to really map a large tissue or an organ. So we have to think about alternative strategies to make this more efficient. That's why we developed this method called SHAF, and using imaging methods that people will have more access to, and then it's being routinely done in the clinical settings. That is to use the histology pictures. And there's a lot of archived histology pictures in clinical settings. So SHAF has two different modes. And in one mode, if you have paired data with HE staining with spatial transcriptomics, you can use the paired SHAF. But in most of cases, we just don't have this paired data. But we can also use the second mode of the SHAF, which is the unpaired SHAF. So you have single-cell RNA-seq from one tissue or one slice. You have HE from another slice. So we can use both, and then we train the model on different kind of histology images, and then generate the spatially resolved single-cell RNA-seq data. So let me just give you a few examples of how this might work. And imagine with your input, this is a ER-positive breast cancer. With your input with histology pictures, and this is the single-cell RNA-seq data generated by this HE staining, and we did a clustering and annotate different clusters. But the model never sees the ground truth. So this is the ground truth measured by 10x cesium. So this is in that cell type level that you can do a pretty good job. So you can just take an HE staining and then predict the spatial transcriptomics. And then you can also do a reasonably good job for marker genes. Just to give you one example, this is ERBB2, and it's a marker gene, and you can see that the predicted gene expression and the ground truth looks very, very similar. But this is just, at the best cases, and then you can do pretty well at marker genes, but we're far away from how accurately we can predict all the genes from just the HE staining, because the information encoded in the HE, it's still limited. You have 3 RGB colors, right? So it's still limited. So that's why we wanted to even push this further. And as first, I give you the example of how we can generate single-cell spatial transcriptomics data from the paired data. But in reality, most of the data is actually unpaired, right? So this is just one example that we recently released the single-cell multiomics data from the human placenta. And then we use the same strategy, but we just don't have the paired HE and spatial transcriptomics from the same slides. So the single-cell RNA-seq is from one slide, HE is from another slide. But we train the model again, and this is a new HE slice of a human placenta, and it's actually very, very big. And this is the small region where you can choose your favorite spatial transcriptomics measurement, but this is the scale that you can actually map with the current spatial transcriptomics methods. But with these methods, we basically generate the spatial transcriptomics data from the whole HE slide, and this is what the cell type look like, and this is the expert pathologist annotated results. We also have the quantitative metrics to show how this works. This is all good, right? And then you can do this with, again, with marker genes, and then this is the front row, like the first row shows you the marker genes predicted by Schaff, and this is measured by the ground truth measurement using star map. But this is just, the best results we can see. But actually, in many of the other, lowly expressed genes, these are not there yet. So how can we improve this? How can we even just, get more information from the imaging side, and maybe you can actually capture the subtle differences in the gene expression? So that's why we wanted to extend this effort to different other imaging modalities that, chemists have been using this for a long time, but biologists that haven't been using this. For example, this is the infrared imaging. It's another type of chemical imaging, but it gives way much stronger signal-to-noise ratio. It measures also the absorptions of different lights and then the interactions between light and the molecules. But it gave you 500 channels per pixel. And this is extremely fast. So take a picture like this. It only takes us 10 minutes. And this is label-free, so you don't need to buy any reagents. So which means the running cost can be reduced significantly. Of course, you still have to buy some coffee for students so that they can take the pictures. And this is on the other side, and I think people in this campus, they may be also doing this. This is quantitative phase, which is very gentle and it's very fast. You get an idea of the 3D information of cells. So these are just like another 2 different kind of imaging modalities that we're trying to push. But so far that I show you that you can use easy image measurement to generate the spatial transcriptomics data. But you will also ask, can you do the reverse direction, like the OpenAI's DALI, or even if you have the time information, that would be like a video, like a Sora from OpenAI. So the short answer is yes, you can do this to a certain degree, right? So that's why we developed this method called Genvenci, generative Da Vinci, so to show tribute to the greatest painter. So the input here will be your single-cell gene expression, spatial transcriptomics data, and we developed this diffusion model, and then it's a gene-to-image translation model that we can actually generate different kinds of cellular or tissue pictures, including the HE staining, DAPI, immunofluorescent, neuromorphology, I think it might be interested to many people here, and then even the electron microscopy images at the single-cell resolution and also multi-cell or even tissue or whole slide level. But let me just go through like 2 examples. The first example is that we can actually generate or translate the gene expression into neuromorphologies. This is what the patch-seq, the staining of the neuron, what it looks like. I saw some pictures outside like this too. And this is the model generated. We asked the model to generate multiple different kind of pictures, and then it's just to show you what it looks like. So this is what the ground truth look like, and this is what the model generated. We also compare that with vanilla baseline models because we couldn't find similar work that can do this kind of work for a benchmark. But I think what's more exciting for the audience here is we also demonstrated that you can actually generate electron microscopy images. Of course, this is not as good as AlphaFold or DALI because we only have 900 cells in training. So this is the best data that we can actually have. And this, again, the first column shows you the ground truth EM pictures, and this is what the pictures generated from our model, and then compared with the baseline model. We also have quantitative matrix to show how similar these two pictures, the generated picture and the ground truth pictures. So again, I just give you this, introduction of how we can think about the translating different kind of languages to have a multimodal view of the cellular states. But as a biologist, we wanted to study perturbations, right? We wanted to perturb the cells and map different things at different modalities. So that's why we developed another method called VIBE-OT. VIBE here represents vibration. So we measure the vibrational energies inside of a cell. So that's enabled by the chemical imaging, either Raman or infrared imaging. And then we use a mathematical model, optimal transport. Also, Caroline introduced this to the field, and to combine high-dimensional, label-free chemical imaging with perturbations. So we showed this that with 19 different clinical approved drugs, we treated with one cancer cell lines. And what optimal transport can do is to predict the drug response at the single cell resolution. So basically, you have an unperturbed state. You have and also a perturbed state. So the optimal transport can actually map this distribution from control and then and then the perturbed state. But because we're using the high-dimensional chemical imaging, I give you the two examples, the infrared and Raman imaging. If we do Raman, which means that it's also compatible with faster version of Raman, which is the stimulated Raman SRS imaging as well. So you can do this to get a spectrum information at a high dimensionality. That this is per pixel, and then you average and aggregate the different pixel information, the spectrum information, and this will enable us to actually model or uncover the biochemical properties inside of a cell. And I show this, we are only doing this on imaging, but at the same time, we're also coupling this with the spatial transcriptomics, and you can build this the same way that I showed you before. But I just wanted to give you one example that from the spectrum information you can see, just by eye, you look at the shapes of the spectrum, and then the different drugs, which we didn't actually give the model that they don't know the labels. They don't know what this drug does. But it turns out the drugs have similar mechanisms of action here. They have similar shapes of the spectrum pictures. So which means that it turns out all the three drugs are protein synthesis inhibitors because you can actually extract the information from this high-dimensional chemical imaging. So in summary, I think we just show that you can do this different kind of languages, translating between different kind of languages, and then we've been doing this at a smaller scale in a small academic lab that you can do different kind of modalities. But I hope that this could be a community effort that maybe in the future we can imagine that we can actually build a Google Translate of different kind of languages in biology so that you can actually have a multimodal view of the physiology across different living systems. But there's one problem. There's just no database that actually enable us to do this. So that's why I think maybe this is worth a conversation, especially for this audience, that how can we talk about some community effort to build this organized human knowledges across the different kind of modalities so that you can actually have a AlphaFold moment for different kind of data modalities, or maybe like a scale AI, that kind of thing for biology. So in summary, I mentioned that biology is multimodal. And you can also translate between different kind of languages or different modalities. You can also perturb each of these modalities and measure the impact of different other modalities. So maybe putting all these different pieces together, then we can actually study the actual human physiology across scales and modalities. So thank you.
I think we have time for one question.
Jian, this is cool. I'm interested for the gene
Vinci.
Yeah. So actually, for that to generate image, do you all need all the information from gene expression, or actually you know there is a key cohort of the genes actually have the most predictive power or generative power for imaging?
That is a wonderful question. We're not there yet. We've done some ablation studies to just like remove some of these genes to see how well you can generate the pictures, so that you can actually figure out what kind of gene modules might be highly correlated to the spatial structure of the cells. Because of the data, I don't want to just blame data, but it's actually the reality that we just don't have enough data to actually have all these perturbations, so we can actually find out what kind of modules are actually causing these morphological changes. Yeah.
Let's thank Jian and